So we've had a dose of fairly harsh reality. I think we now need to be cheered up, and I can think of nobody who can do that better with conviction. And then our next speaker, Dr. Joan Merril, who is going to discuss the topic of future trials of lupus. Can treatment development be any less risky? Joan.
I've been told that it costs up to 20 million US dollars, not Canadian dollars, to do a phase 2 trial, and sometimes hundreds of millions of dollars to do these large international trials. This means that all of these failed trials are making people in industry very queasy. Future trials are going to have to be less risky. Is there a way forward? These are my disclosures. You know, you've been hearing compelling evidence from both Ron and Rich that we're beginning to understand ways to at least look back at data from trials that we've already done and identify subsets of patients where we could have seen a better difference between treatment and placebo. This is true for failed trials. This is true for trials which succeeded but maybe had somewhat disappointing endpoints. Suddenly they look a lot better when we take subsets of patients that came into the trial a little bit sicker. And the case has been made that there are issues with what sorts of background medications we're using, how many background medications to use, when should they be tapered. And so it makes sort of sense that this case arises from the fact that the sicker patients are less likely to respond to increased background medications. Now, when you do trials of sicker patients, which is what everyone seems to be jumping on the bandwagon to do now, you can include more background meds, and in fact you must include more background meds because these patients are ill. But you're now risking increased infections in these clinical trials, which may raise a barrier and actually has done in some of our recent trials to the further development of a drug. You certainly are addressing an important unmet need in lupus, and by sort of restricting the use of belimumab to sicker subsets of patients, many of the regulatory agencies around the world think they're doing us a big favor because they're saving money for the world and we're only going to use these drugs in the people who need them. But this results in a limited market. And if I owned a very large international pharmaceutical company, I might not be interested in developing drugs and spending millions of dollars to do so for a limited market. But more profoundly for us as physicians and for all of us in this field, a limited market means a limited number of patients with unmet need. And if those of you who came to the Lupus Academy will recall that there was a great deal of discussion there about what Michelle Petrie likes to call middle lupus. These are patients who may not have such severe disease, who may get partially better all by adding steroids to background medications and somehow clog up and cause noise in our clinical trials. But these patients are going to die early, and they are going to accumulate increasing damage over time, and they actually represent a huge unmet need. And so if we could do trials in which there were less background medications, using these patients who might be a little less sick, perhaps we could see a difference between treatment and placebo, resulting in trials with fewer infections, resulting in trials that would be easier to recruit around the world because of all the competition right now for the few patients who actually qualify for clinical trials. And most importantly of all, there would be a bigger market for pharmaceutical companies to pursue, and we would be addressing a much wider and equally as important unmet need for our patients. And so that's my opinionated introduction. I will now proceed to show you some data. Now, for those of you who are at the Lupus Academy tomorrow, I apologize. I'm going to be reprising some of these slides in the beginning, but I will get to some new stuff at the end. There's evidence from a number of clinical trial reassessments that, in fact, less background medication could lead to more interpretable results. Here is on the left, the same picture that you saw before of the Explorer trial, showing that everybody got better on steroids and nobody flared up later, and so there was no difference between the placebo group and the people in orange treated with rituximab. But if you look just at the subset of patients who received methotrexate as their background therapy and eliminated those who received azathioprine or mycophenolate, what you found was that at the end of the trial, in fact, the people on rituximab stayed well, and the placebo group did start to flare up. Now, I don't think any of us think of methotrexate as an ineffective drug, but one could make the case that it's better for arthritis than many of the other manifestations of lupus. So these patients have had a chance to show us that they really are not going to stay well once the steroids are tapered off. Now, this is actually the slide that was referred to in response to Joe McCune's question about the background medications and the use of cyclophosphamide with biologics, especially with abatacept. This is actually a trial of ocrelizumab, and it's a cautionary tale here because ocrelizumab is a humanized version of anti-CD20, so it's similar to rituximab in its mechanism of action. But in fact, you can make a case from this slide that it is better to use cyclophosphamide as a background therapy. But you could also make a case, which I'm about to, that that would be unethical. If you look at the results on all patients, there was very minimal difference between the placebo group in yellow and the treatment groups. And just to help you out here, I think we should just look at the green line at the end, which is sort of a combination of all the results. So you can see that there is very minimal difference between the placebo group in yellow and all the and the treatment group in green. If you look at the people on the far right who received mycophenolate mofetil as their background medication, equally there is really not so much difference. If you look overall at the people who received the Lupus regimen, there seems to be a bigger difference. But is it because the treatment group is doing so much better, or is it because the placebo group is doing so much worse? And so it may not be so much a question of synergy in all cases as it is a question of, can we decrease the strength of the background medication so that we can perceive an impact of a biologic? Remember, there must be a ceiling for what percentage of patients can respond to a biologic in the first place. These are targeted agents, and not all patients are alike. And so that's something to keep in mind. But now would it be ethical to do an international trial using the Lupus regimen as a background? Because the most compelling take-home message from this slide, I think, is that mycophenolate mofetil alone performed much better than the Lupus regimen alone. In the belimumab trials, you've seen these data already. One of the trials had a very sustained and clear difference between the placebo group in yellow and the intermediate dose in purple and the higher dose group in blue. One of the others also met its primary endpoint but had a little bit more disappointing results. And although we don't know the reasons why these two trials didn't perform exactly alike, there is a factoid that we should keep in mind, and that is that more patients in the trial that looks a lot more discriminatory started the trial taking steroids. And in fact, data shows that many of those patients were able to taper down to low levels of their steroids. Whereas in the other trial, more patients entered on background immune suppressants. They were allowed to be increased during the trial, and although they were allowed to be decreased, we really don't know what percentage of patients actually did that. So there is a possibility that here, too, we're seeing an illustration of better discrimination with less background medications. Now, of course, belimumab targets a drug called a cytokine called BLyS, or BAFF. So BLyS, of course, is not at the same level in all of the patients. Our patients are not all the same as each other. But I want to point one more thing out to you, that when we entertain these clinical trials of targeted biologics, are we asking the right question when we allow disparate background medications to be used? Because how do I know what percentage of these differences in the degree of BLyS in lupus patients is due to their intrinsic biology, and what percentage is due to the background medications that they're on already? So these drugs that they're on in the background could be interfering with the mechanism of the drug we're studying. They could be accentuating the mechanism of the drug we're studying. And there's no way to stratify and understand all of that. So therefore, is it possible to do clinical trials where we eliminate background medications entirely? This was the first foray that we made into that, with a very small trial of 27 patients, of whom 13 received placebo and 14 were given in a blinded fashion, mycophenolate mofetil. It was a study of lupus arthritis. Patients could have multiple things going on, but they all had to have arthritis. And you can see that in this study, even though the response rate to mycophenolate mofetil was quite modest, it was about 30%, there was a statistically significant difference at the end of the 3-month period of assessment because the placebo group had no responders, because we took away their background medications. So patients came in, they took away their azathioprine or their not their mycophenolate, they couldn't have been on that for this trial. They took away their azathioprine or their methotrexate. They received a short burst of steroids very quickly to wear off, and 3 months later there were no responses in the placebo group, and now we could ascertain an impact of mycophenolate mofetil. And because we took away the background medications, we could start to look at some interesting things about the biology of the patients. And so it turned out that if patients came in with a high expression of a complement receptor on neutrophils called complement receptor 3. And here I have to thank Jill Bion because I was her fellow and she tormented me in the lab for 2 years studying this molecule. So I was prepared when we did this trial to do this study with it. By the way, you've heard of a gene that's one of the genes that seems to increase risk for lupus called ITGAM. ITGAM actually codes for this molecule. So people who came in with the 5 patients who came in with the highest expression of complement receptor 3, and all of them were more than 1,000 units high, when they received the treatment, it went down. You can see that on the left-hand side of the slide. The 5 people who came in with the lowest expression of ITGAM, when they received mycophenolate mofetil, it didn't go down even further. It rather just sort of looks like it regressed to the mean. And so that was sort of an interesting observation. And then it turned out that if you looked at, after 4 to 8 weeks of mycophenolate treatment, if you looked at patients who'd had no response, they still had relatively high levels of expression of this molecule. And if you looked at those who had a major clinical response, they had decreased levels of this molecule. And in fact, if you simply looked, forget about arthritis, let's go to the SLEDAI and the BILAG scores that are used in clinical trials. If you looked at the high group over on the left, and you looked and compared their SLEDAI scores before they received mycophenolate mofetil and after, these improved. If you looked at those who had low expression, they were not responding at all. And the same was pretty much true if you measured using the BILAG. And this led us to contemplate a real study of this placebo group, because that's what I think is the first step to designing a clinical trial. Do we understand a little bit about the background biology of our patients, and is it really as heterogeneous and confusing as we all think it is? And so 100 active lupus patients were proposed to be enrolled. In fact, it ended up being 103. We've now completed this trial. 41 went into a sort of a prospective study in which their background medications were withdrawn, given a short course of steroids, followed to time to flare. And then another 62 gave one donation at one point. This allowed us to study patients who were a little sicker, but we did not withdraw their background medications. I'm not going to give you the top-line results from this study. It's just completed. We're still analyzing it, and we're hoping to submit it to the ACR. But I am going to show you some of the preliminary data that came out looking at the first 54 patients. Again, 20 patients had completed Group A, and 34 had also given samples to us at that time. And so you can see for the patients who went into this prospective study, when you give them steroids and you withdraw their background medications, they don't suddenly flare up. They all get better because we know steroids work. So we're looking here at the first visit, the visit that was designated as the improving visit, slightly different for each patient. Sometimes it was at 2 weeks, sometimes it was at 4 weeks. And we're doing sort of modified SLEDAIs and BILAGs because when they improved at 2 weeks, we allowed the instrument to go back 2 weeks instead of a month. That's just a little modification we had to make to study these patients. And then you're looking at them at their flare visits, and some of them were as early as 3 weeks. Okay? So you can see the SLEDAI score goes down and then up. The BILAG score, known to be a little more sensitive to change in the SLEDAI, goes down even more and up even more. And if you look at the time to flare in these patients, the patients in red had more severe disease activity and they flared a little sooner, but the bottom line is every single patient in this trial had flared up again within 6 months, and I will just give you, I have to modify that because there's one patient out of the 41 who didn't, but most patients have flared within 6 months. Now we can start to look at the biology. And what's interesting about this study is you can actually look at the biology on the background drug versus the biology after they're flaring up again. Same patient, less heterogeneity, serving as their own control. Now they're not on the background drug. So let's look at BAFF, which is also BLIS, which is the target for belimumab over here on the left. It's the only one I'm going to focus on here because we don't have much time. You can see in red the levels of this BLIS in patients as they enter the trial. And at the top, over on the left, this is a patient with pretty low levels of BLIS in red. And you can see that it gets a little bit better and it gets a little bit worse as the patient gets into their clinical improvement and their worsening improvement. And those big dips you see are their SLEDA and BILAG scores in green and blue. Here on the bottom, though, there's a patient who came in with very high levels of BLIS. And you can see that this patient dipped down dramatically, but never even reached the place that the first patient had been on in terms of the level of the BLIS. And again, popped up again when they flared. So steroids certainly seem to have some kind of impact on BLIS while they're having an impact on the clinical improvement. But here's what we don't know and what we would need to know to really be brilliant physicians in the use of belimumab. Do we know whether the patient on the bottom is even a candidate for BLIS? I don't think we know that yet. We know steroids can make the BLIS dip and that goes along with improvement, but that's an association. It's not necessarily related, okay? Is that patient at the top getting enough treatment? Would they flare a lot later if we'd gotten their BLIS down to a lower level? This is another thing we don't know yet. And there's more to it than that. What if it's not all about having high BLIS, but it has to do with how BLIS is being received in the cells where it's targeting the B cells? Are there receptors on those B cells that make the BLIS more potent so that even that low patient needs to have their BLIS signal dampened? We don't know the answer to that either. But we're beginning to see the variables in the patients now, and we're beginning to understand what some of the questions are in making us better doctors biologically. Now, all of you are aware that interferon alpha is a very big story in lupus. And it turns out that we, like many others, seem to be finding in adult lupus patients. You'll see in brightest red, you can color these things any way you want, but the red guys on this particular picture are the people with low expression of the genes that are triggered by interferon alpha, and the people in green are the people with a high expression. So you can see that about half the patients have high expression of genes that are triggered by the activity of interferon alpha. So it turns out, and unfortunately we were not the first people to observe this, but we're about the second or third. If you look at the impact of the interferon signals on the BLIS levels, the control patients are over there in black. If you look at the people who have low expression of interferon alpha inducible genes, they do not have BLIS levels much higher than the controls. It is the people with a high interferon signature that have the high BLIS levels. Suddenly, we're beginning to wonder if lupus couldn't be a little simpler than we thought. And so if you look at the expression of interferon pathway genes as being either high or low, and you see here in the first column where it's white on top and gray on the bottom, we're looking at the high interferon patients. In the second column, we're looking at the low interferon patients. And now we're looking at the expression of a whole bunch of genes, and what you see is their patterns that are reducible to whether you're a low or a high person. So if you're a low person on the top of this graph, there's a tendency for a decrease in the genes you see over there in their expression. If you're a high interferon patient, there is a tendency for an increase in this other set of genes. But when the high has something that looks statistically significant versus controls, it is not shared by the low people. And when the low people have something, it is not shared by the high people. And so, in thinking about the impact of medications on expression levels of these genes, which may give us some biologic understanding of how to design our trials and which of these medications we ought to let into our trial, because we would be designing drugs that hit a certain pathway. We want to know what these background medications are doing to these pathways. It turns out that if you look at antimalarials, you see a statistically significant impact in the high interferon group on the expression of one gene over there that's not shared by the low group, and you're seeing an increased expression in the low interferon group that's not shared by the high group, and this whole chart basically is saying the same thing. No matter what drug you look at, their impact differs depending on whether the patients are interferon high or interferon low. So we've heard some solutions for clinical trials today. I think that both Ron and Rich stressed very importantly that we can be successful if we look at the more 6 subsets of patients, and made some important comments about how we design the endpoint measures for the clinical trials. We have huge problems with large phase 3 trials. They're expensive. They raise horrible training and recruitment issues. We don't have enough patients around the world in trained trial sites to be doing these trials at the moment. And if you go into a large phase 3 trial not quite knowing what you're doing, look at the risk that we're exposing so many patients around the world to. So we need solutions for smaller, more efficient trials to help us to understand what exactly we're doing before we go into these huge enterprises around the world. We certainly could restrict these studies to sicker patients, but there's a problem there. It's really not addressing the unmet need, the huge unmet need around the world of lupus patients. And if you expect pharmaceutical companies to shoulder the burden of developing trials for a tiny subset of lupus problem patients, I think we should dream on. So another solution to meet the large unmet need of these patients who are accumulating damage, morbidity, and have early mortality from lupus, we could entertain the idea of trials in which in various ways we might decrease their background medications and think very hard about the impact of these background medications in our selection of what we allow in on the actual drug that's being tested. In short, we need to nail the biology a little better in our clinical trial designs. Thanks.
Questions for Joan, please, while people go into the microphone. So thank you for that. I entirely agree that more intelligent trial design is essential. I wonder if I could suggest something to you. It's clear that B cells are very integral to the development of lupus, and we know that drugs, anti-CD20 drugs, will be very good at eliminating those cells. But we also know that B cell activating factors will help to their return. Would it therefore make sense, do you think, to design a trial where you used a B cell depleting agent, waited a while, and then used something like atacicept, which would block 2 B cell activating factors to prevent the return of the disease?
Well, in fact, I'll take that one step further and I'll up your ante. It turns out that when you give rituximab, the levels of bliss are markedly increased. And although you could say, who cares about that? There are no B cells there for it to activate. We don't really know that. We don't really know all the places there are bliss receptors or all the places bliss may be acting. And so I think, Betty Diamond in the United States is entertaining a trial where she is going to actually give those 2 targets in combination. I think waiting for her results will be very interesting.
Thank you. Quick question, Central.
Yeah, Frédéric Couchot from Brussels. Jill, as you might anticipate, I fully disagree with your interpretation of the BELONG data. When you say that the major message in the BELONG trial is that CellCept is superior to Lupus, I think that the trial, first, was not designed at all to prove superiority or inferiority of one regime against another. But more importantly, you probably know that patients randomized into the Lupus group received much, much less steroids than patients randomized in the MMF group, and in particular, less steroid pulse therapy. And that might be an explanation why these 2 groups are slightly different.
I do agree with you about that, and I'm sorry if I sounded at all cavalier. In fact, the Lupus regimen is very effective for many patients, but one of the key differences between these trials was the large percentage of patients from other parts of the world that were participating in the BELONG trial. So I think, time will tell where it is best to use each regimen. There is no doubt that the Lupus regimen is effective, at least in certain subsets of patients where it has been properly tested. And you are right, that was not an endpoint that was pre-specified, and therefore we cannot draw conclusions from it. You're quite right about that.
Thank you, Jill.
Is it Joe?
I guess that lupus trials are afflicted with a number of problems which you mentioned, but two problems you didn't mention. And I wonder what you think, whether they're not problems and therefore you didn't mention them. One is the heterogeneity in outcomes, which makes it so different from RA, as was pointed out by Rich before. But the other, maybe more important aspect, which was also seen in some of the studies with ANA positive, 25–30% not having ANA positive activity and being called lupus. The other aspect is the vast heterogeneity of the disease, and everyone tries to put in this heterogeneous group where we don't even know if the skin disease has the same pathogenic pathways as the joint disease or the renal disease and the hematologic disease and so on. So wouldn't it be important to focus more on homogeneous groups of patients, regardless of what you call them? Arthritis patients, patients with particular skin disease, because bullous skin disease will be different from the regular rash. So wouldn't that be more helpful in order to understand and find new therapies?
Now, although we don't really know how biologically homogeneous these patients are who present with the same disorder, but in fact, one example of where they may very well be is skin disease. So I'm a great proponent of thinking about doing early trials in skin disease to really nail, and what a wonderful opportunity that is also to do skin biopsies on these patients and really understand what this agent might be doing.
Can we have one last question over there, please?
Eric Moran from Melbourne, Australia. Joan, I really applaud your small study where you've ventured into withdrawal of background therapy, and I strongly agree with you that's somewhere we need to go as we look for better therapies. Do you have a sense that one day we'll know how long it's possible to withdraw background immunosuppression for in a given group of patients, and that might influence our ability to design short trials? Are we getting that information?
Yeah, if you made your primary endpoint, say at 3 months, we've already shown that it's reasonably safe to do as long as you're selecting your patients. I would never put an active nephritis patient in a trial like that. And I have certain TIPI patients who I'm scared of, who I would never put in a trial like that. So we do have to depend on some sort of rigorous thinking by the investigator. You could put your primary endpoint at 3 months, and you could allow patients to be treated after that. What's the difference? You're having the same noise as other trials, but you've managed to get your primary endpoint quite clean. Or you could even allow treatment at any time, but call that person a permanent non-responder. So I think it's a feasible trial design. In fact, I do know of 2 phase 2 trials that have used this design. One of which is being presented here. Actually, it's completed. The rontelizumab study that Dexter Kennedy is going to be presenting on Saturday did use this trial design. And one of the things that allowed was that they found a very small subset in their trial where they saw an efficacy signal, which they could not have predicted, the surprising subset, and which never would have been seen if they hadn't used this trial design, in my opinion.
Thank you. Professor Vasconcelos will have the final word.
Well, I just want to make the point that taking off background medication in placebo arm can solve trials problems, but does not solve patients' problems. We do need better drugs for the patients. Thank you very much.